Коллеги, сейчас у нас будет очень интересная на самом деле лекция, касаемая той группы пациентов, которые, ну, как сказать, ну, практически у всех, наверное, вызывают вопросы, причем вопросы очень серьезные. И зачастую именно такие пациенты воспринимаются совершенно по-разному в разных лабораториях, потому что, в общем-то, действительно, это лейкозы совершенно неоднозначные, очень сильно отличающиеся, допустим, и от лимфобластных, и от миелоидных лейкозов. Зачастую очень непросто понять все-таки, что это за лейкоз, а уж тем более понять, как его лечить. И поэтому мы попросили Марию Кристин провести нам лекцию на эту тему. Это, я думаю, что будет очень важно, интересно и очень полезно. Maria Christine.
Okay, good morning, everyone. I'm glad to be with you. I would prefer to be in Moscow, but at the moment I'm sitting in front of my computer and I will talk to you from my room and at home in Nantes. Thank you very much for the invitation to take part to this event. Sorry, and we're going to talk, as you know, about mixed phenotype acute leukemia. This is my disclaimer. And before we start, I think it's important to give some general considerations, because if you want to look at MPAL, you need to know what's normal hematopoiesis. And I guess, I wasn't there, but I guess that Michael also told you that before. So if you want to look at normal bone marrow, you have to work in a sort of systematized way. When you look in the literature, there are many, many ways to look at normal bone marrow, but I would like to present you what we published almost 10 years ago now, trying to find a systematized approach for flow cytometry of normal bone marrow. This was a study that we did with 4 and 5 color cytometry. And what we thought would be that it would be important to first delineate the normal maturing cells of the bone marrow. And we let's try to have a positive identification of those mature cells in the bone marrow and assigning them a color code. You can, of course, use any kind of color code that you wish. I'm just going to show you what we thought and that I'm going to use throughout this presentation. So we decided that a good way to identify monocytes was CD14 and CD11b and to color them in green. A good way to see granulocytes that we put in red was to use CD16 and CD11b, which also allows to look for NK cells and B cells, but we'll use also side scatter to be able to see the granulocytes. And as you know, CD45 is especially bright with a low side scatter for lymphocytes, and we put them in magenta. Then we looked for some help from George Boyle, who's a mathematician, English mathematician, and we prepared a Boolean equation to make a Boolean gate, including all the cells, but not monocytes, not granulocytes, and not lymphocytes. And that left us with a sort of triangular area where all the maturing cells were, and we call that the Bermudes because of the Bermuda triangle where you can easily get lost, and that's the case also when you work in flow cytometry. So if we look at a cartoon of what this does, if you first do a forward scatter, side scatter, and then use CD14 and side scatter, you will see your monocytes, and you can put them in green. If you use CD16 and side scatter, you will see your granulocytes, and you can put them in red. If you look at CD45, and in this instance 11b, you can see that these cells are your lymphocytes. And the good idea, and I heard a little bit about CD45 a minute ago, is to backgate that on the side scatter CD45 gate, which gives you the monocytes, your granulocytes, your lymphocytes in the Bermuda area, and then you find the erythroblasts, which are CD45 negative, as you know. If we do that in a real life, here you see a normal bone marrow with this forward side scatter scattergram, and then you do the CD14 side scatter, and you can get your monocytes. Then you do the CD16 side scatter, you see your granulocytes. You do the CD45, CD16 in this case, you see lymphocytes, and this is what you get as what we call the cartography of the normal bone marrow, because it really looks like a map where you can find what the populations are going to be. And for instance, I used to say that if you look at this as if it were Europe, you would have the granulocytes in Sweden, the monocytes in Austria, the lymphocytes in northern Italy, and the Bermudes around the Pyrenees down there. So it's really a map, because anywhere you're going to look at your populations afterwards, you will find, because of the colors, in which part of the map you are, and if you do the backgating, you can confirm at which population you're going to be working with. Then let's move to leukemias. You know that the origin of acute leukemias is clonal hematopoietic cells. One hematopoietic cell is going ill and it proliferates and it's giving you the leukemia. And these cells are either blocked at a given normal stage of maturation, and then they display a normal immunophenotype, or they can express aberrant markers. And we call that immunophenotypic patterns, leukemia-associated immunophenotypic patterns, or LAIP. And that's, of course, probably what Michael talked about in his previous talk about MRD, because this is what you're going to look at in MRD. These LAIP, they are not normal, and they can also be called different from normal. And that's another concept that is more and more used and recognized, because especially with the tools of automated unsupervised assignment of cells, we are also working on this kind of tool. Then you can compare normal bone marrow and diseased bone marrow, and you will find population that are absent from the normal bone marrow, but that give rise to the leukemia. And this is called also DFN, and DFN and LAIP in most cases are almost the same. So the diagnosis of acute leukemia will be needed to confirm the lineage. You can get an ID by morphology, but flow is going to confirm the lineage of the cells that are growing. It can be used for a classification, and we have been publishing that a number of years ago. It can be used also for the identification of LAIP that are going to be useful later on to do MRD. And of course, and that's what we're going to talk about today, they are important to identify what was initially called biphenotypic acute leukemias, which is now called MPAL. So to do that, you need an extensive immunophenotyping panel, and it will allow you to find out whether your cells are myeloblastic or lymphoblastic. Myeloblastic leukemias can be classified according to the maturation stages of myeloid cells. There are no well-defined stages of maturation. There's a sort of continuum. And what you can do is to consider that this not defined stage of maturation depend on the fact that there's a high level of production of cells in myeloid cells. You make about 20 billion neutrophils every day, and they're going to live only one day. So there's not very much specificity in the recognition of antigens. These cells are going to recognize many danger patterns, but they do not need to have a very exquisite specificity. The major antigen in the myeloid lineage is myeloperoxidase, and there are also other myeloid markers. And of course, the first one is myeloperoxidase. This is this homodimer here, but you also have CD13. CD13 is an ectopeptidase. It is a type 2 molecule with an H2 in the cytoplasm, and it's got here 2 enzymatic sites. It's almost the same molecule as CD10 that is expressed on neutrophils and also, of course, on maturing B cells. And these 2 antigens are very important in the physiology of the human body, because you have a lot of them in the kidneys, where they are going to work with the angiotensin conversion enzyme to make it functional. And you also have it in the enterocytes in your gut. And these molecules in your gut are cutting proteins, are cutting off 2 amino acids at the end of any protein or peptide. And in fact, it's very useful for you to absorb the amino acids when you eat some protein food. So you will never see, probably, it would be very dangerous to give antibody therapy with anti-CD13 or anti-CD10 because of this physiologic expression of these molecules. Excuse me. Another important molecule, of course, is CD33. This is a molecule of the immunoglobulin superfamily with adhesion properties. And this one is really specific of the myeloid lineage. CD117 is also a molecule of the superfamily, the immunoglobulin superfamily. It's got a kinase activity in the cytoplasm, and it's also the stem cell factor receptor. It's expressed constitutively on mastocytes, mast cells. Then you find CD14. CD14 is a GPI protein, which is just picked into the membrane with no transmembrane and no cytoplasmic end. It's got a lot of leucine, and it's also the LPS receptor, so it's going to recognize bacteria very easily. CD11B, I just put that one, but there are many molecules of this kind on the myeloid cells because these are integrins. This is an integrin alpha chain. It goes with a beta chain. And these are important because they are in some way the legs of the cells. This extracellular part is going to bind to the extracellular matrix so that the cell can stick on the extracellular matrix, and in the cytoplasm, it's very much linked to the cytoskeleton of the cell. So it's really a mobile or mobility element that's going to allow the cells to move to the place where they're expected to be working. CD15 and CD65 are two sugars with undefined roles, but for us, they are very good markers of the granulocytic lineage and somehow also on monocytes. CD35 is a complement receptor, which is seldom used in flow cytometry, but it could be a good marker of these cells. CD36 is much more used. It's a thrombospondin receptor. It's present on monocytes, but also on platelets and on nucleated red blood cells. So it's a marker that's going to give you a lot of information when you use it. CD16 is a receptor for immunoglobulin constant fragment. It's going to be extremely useful for the myeloid cells in this GPI form with no transmembrane and no cytoplasmic part, and it's going to help these cells to catch immune complexes, the antibodies of the immune complexes. This part here, this form with the transmembrane part, is present on NK cells. And last but not least, there's this very tiny molecule, CD24, which is almost the same as CD52, which is the target of the alemtuzumab, a monoclonal therapeutic antibody. It's only a few amino acids, a GPI anchor, some sugar on it, and it's a very good marker of B cells, but also of neutrophils. So what happens during the maturation of the cells? It starts, of course, in the bone marrow. And in this bone marrow, you will find a stem cell, which has the characteristics of bearing CD34 and for unknown reason HLA-DR. But of course, CD34 you heard about before and you know about its very earliest lineage marker of the leukocytes. And then when the cells decide to become myeloid cells later, they will express immature markers, and they are immature precursors expressing CD117, CD13, CD33, MPO, and for very transient time, CD7, which is a pan-T cell marker. Then it's going to get out of the bone marrow and reach the peripheral blood and also tissues where needed, and they become differentiated cells, which are expressing a panel of these different markers. And of course, at this stage, you can recognize them also very well by their morphology, from the monocytes to the neutrophils, basophils, and eosinophils. And these markers are going to be shared by these different stages. So now if we move to lymphoblastic leukemias, they can be classified according to the maturation stages of the B or T lymphocytes, of course, and there will be many similarities between the two lineages. The first one is that these cells have a very early commitment. It's a very soon in their life that they decide that they're going to be a B cell or a T cell. They also share the fact that they're going to rearrange their DNA to prepare the very specific antigen receptors that they're going to bear, the BCR and the T cell receptor TCR. And because this rearrangement is done in the bone marrow in a sort of without any contact with the antigen, it's just by chance that they're going to make a receptor, then they're going to make autoreactive receptors, and these cells need to be removed. So there's a huge step of selection where the cells who have a BCR or the TCR reacting with autoantigens are going to be removed from the body. So if we look at the markers, the B lineage associated markers, of course, the most important is the BCR, which is this immunoglobulin on the surface of the B cells with 2 heavy chains in blue, 2 light chains in yellow, a transmembrane part, and only 3 amino acids in the cytoplasm. So that's not enough for this molecule when it has recognized by its FAB the antigen that is going to trigger the immune response. It's not enough to trigger it. So it has to work with 2 other molecules of the immunoglobulin superfamily, which are called CD79A and B. And these are carrying in a long intracytoplasmic tail immunoreceptor tyrosine-based activating motifs that are going to provide the activation of the B cell. And of course, we will see later that this is the earliest step to define a B cell. Then you have CD19, also a member of the immunoglobulin superfamily with a long tail with also ITAM motifs. CD22, which can come with 5 or 7 domains of the immunoglobulin superfamily, and which is carrying immunoreceptor tyrosine-based inhibitory motifs that are going to balance the activation provided by the items. Then, of course, the very famous CD20 with its 4 transmembrane parts and these 2 loops outside. We use it, of course, as a target for immunotherapy, and it has completely changed the fate of patients with lymphomas, but we still don't really know what it does. It's probably a cation channel, but it's not formally determined what the function of this molecule is, but it's a very good B-cell marker. CD21 is the Epstein-Barr virus receptor. CD23 is a lectin with a part here that is going to recognize sugars. It's also a type 2 molecule with the NH2 inside. And when it's cut out, it's going to be a B-cell growth factor. Of course, it's going to be very useful to identify chronic lymphocytic leukemia. Here we find again CD24 that we saw before, because it's also a very good B-cell marker. And CD38 is going to come later on activated B cells and also on plasma cells, again, a type 2 molecule with the NH2 in the cytoplasm. So if we look at the maturation of these cells, it of course will take place in the bone marrow. And in the bone marrow, we start again with the stem cell, which is carrying CD34. I didn't put back actually DR, but it's there. And if you look at the genes of the heavy and light chains of immunoglobulins, it's germinal configuration, like in any other cell in the body. Then the stage pre-Pro-B occurs where you see the expression of CD19 and the cytoplasmic expression of CD79. We usually look at CD79A, but both are there waiting until the BCR is ready to carry it up and put it on the surface of the B cell. Then PAX5 is activated, the gene, and it can help the cell to move to the next stage where there will be a transient expression of CD10, the molecule, the ectopeptidase that we saw before. So that's a very early, but a little bit later stage of the maturation cell. And then the RAG enzymes are going to be activated, and they are going to be able to start to rearrange the immunoglobulin chains. So in the next stage, which is called Pro-B, you see here the expression of CD20, and that's something that's important because it means that when you treat patients with antibodies to CD20, you actually act only after this stage. So you will still have a reservoir of cells that are going to be able to repopulate the patient. It can take a long time, but you still have the potential to make new B cells. And at that stage, it's going to start rearranging the heavy chain gene. You have 2 chances on 2 chromosomes 14. One can miss, then you have the other one. And if both fail, then the cell disappears and doesn't grow much later. Once this rearrangement is okay, you will have the expression of intracytoplasmic mu chain in a way that the cell is checking that this rearrangement is productive and that it can make a mu chain. CD10 is disappearing at that stage, and you will see the appearance of CD21. And some of these mu chains with surrogate light chains are going to come in very, very small quantities on the surface of the cell and start doing the selection stage that I mentioned before, removing the cells that have been rearranged in a way that they are recognizing autoantigens. It also gives a signal for the light chain gene rearrangements, which occur afterwards. And then you will end with an immature B cell or naive B cell, which has rearranged properly one of its chromosome 14 and properly one of its chromosome 2 or 22, giving rise to kappa or lambda chain. And this is going to be set for the rest of the life of the cell. And even when it's going to become a plasma cell, that's going to be the specificity of the antibodies that are going to be secreted. That stage of naive or immature B cells can be recognized because on the surface of the cells, you will find full immunoglobulins, a full BCR, but there are going to be 2 types of it on each cell, expressing both mu chains and delta chains. So you will have IgM and IgD on the surface of these naive B cells. They're going to go in the mantle zone of the germinal centers, and that's also a way for you to recognize this specific type of cells. So once this naive cell is ready, it's going to leave the bone marrow, move to tissues, and wait as a mature B cell that is latent, that the antigen is coming. And when the antigen is coming, it's going to proliferate and give rise to memory B cells, which are going to enhance the possibility to recognize that antigen, and to plasma cells, which express CD38 and CD138, which are going to produce these immunoglobulins in large numbers. For the T lineage, we have also the T cell receptor, which is also unable to trigger very much the activation of the cell. So it has to be linked to the CD3 complex, and especially with these zeta-zeta and zeta-eta chains, which are carrying a lot of item motifs enabling the activation of the T cell. Immature markers are adhesion molecules, CD2, CD5, and CD7. And then the maturation of the cells will give a transient stage of expression of CD1, which looks exactly like an HLA class 1 molecule, even with a beta-2 microglobulin here. And of course, the two major differentiation antigens of the T cell population, CD4, that is going to recognize class 2 HLA antigens on antigen presenting cells, and CD8 that's going to recognize class 1 HLA antigens on every nucleated cell. So what happens is that it starts again in the bone marrow with the stem cell, again CD34, HLA-DR that we don't see here, and the two types of TCRs, gamma delta or larger population alpha beta. 5% of the cells will at the end carry a gamma delta receptor and 95% an alpha beta receptor. And this of course is also in a germinal configuration in this very early cell. Then the cell decides that it's going to be a T cell later on. And what we see, as we did for CD79, is that the cell is going to express CD3 in the cytoplasm. That's the first marker of engagement into the T cell lineage. Then you will see CD7, and very quickly after that, CD5 and CD2. And this will allow the cell, which is still in the bone marrow, to leave the bone marrow, go in the peripheral blood, and reach the epithelial, the thymic epithelium. Then you will have different types of cells, depending on the expression of CD4 and CD8. First, the cells are going to be double negative, then they're going to be double positive, and in the end, they will be single positive for CD4 or CD8. So the double negative one is beginning to rearrange the gamma delta receptor, and it expresses CD1A. That's in the cortex of the thymus, and that's corticothymocytes. Then the cell usually fails to rearrange the gamma delta receptor and starts rearranging the alpha beta, and it's losing the expression of CD1A, and it's moving into the thymic medulla. And it's still double negative. Then the cell is becoming double positive. So it's co-expressing for a transient stage CD4 and CD8 together with the first TCR, the pre-TCR, which is also going to start the selection and removing autoreactive cells. Why does this cell co-express CD4 and CD8? It's because it has to choose whether it will be CD4 positive or CD8 positive, because it has recognized HLA class 2 or HLA class 1 on epithelial thymic cells. In twice more than for CD8, it's CD4 that is winning. So in the end, you will have the single positive naive cells, which have failed rearrangement of gamma delta, a proper rearrangement of the alpha beta TCR, and 2/3 of them are going to be CD4, 1/3 of them are going to be CD8, and of course, CD3 is now seen on the surface of the cell together with the TCR, because then the whole molecule is going to be expressed on the surface of the cell. These cells are going to move in the tissues, and they're going to be waiting for the antigen and then give rise to also clonal populations of memory and effector cells. So that was just to resituate what we are doing with these antigens that we are carrying. And in 2005, there was a consensus in the European Leukemia Net, and that was published in 2011, for the markers that could be useful and would allow for the classification of these lineages, classifications of BAT and T ALLs according to their maturation patterns, and also to identify what we're going to talk about now, which is MPAL. At that stage, we still had not so many markers in the that could be co-expressed and used together. So we sort of stratified the strategy, but of course now with more antibodies available in one tube, it's easy to have 2 or 3 tubes to get all these markers together. So here you find in red the markers for the myeloid lineage that we discussed before. Here you see that you have markers for the B lineage, and here you see that you have markers for the T lineage. CD10 is here in gold because it can be expressed either on B cells or on T cells, but the other ones are usually mutually exclusive. So let's come now to mixed phenotype acute leukemia, or MPAL. They were first called biphenotypic acute leukemia, and you may remember calling them BAL before. And they are characterized by the co-expression of markers of more than one lineage. These are really abnormal cells that have not decided to which lineage they belong. They can be bilineal when you have coexistence of 2 clones of different lineages. That's another type. So you can have two types, real B phenotypic, expressing markers of more than one lineage, or coexistence of two clones. In 2008, it was given another name, the new name of MPAL. And of course, you must remember that it's a rare disease and it can be easily missed when you use two restricted immunophenotyping panels. So the idea of having an extensive panel for immunophenotyping, even when you've seen in the microscope that it was going to be myeloid or that it was probably going to be lymphoid, is that you will be able to identify these MPALs, and we will see why it's important. So in 2008, the WHO classification was very much keen to rely on cytogenetics. So for these specific types of leukemias that were not clearly myeloid or TALL or BALL, it was recommended to have this extensive immunophenotype that would allow to identify acute undifferentiated leukemia. These kind of leukemias have almost disappeared because we have so many markers now that we can, in the vast majority of cases, identify the lineage of the cells, but there still is a little room for them here. And of course, it allows to identify MPALs. And because of this cytogenetic classification that I mentioned, they can be subdivided in three different types. One with the Philadelphia chromosome, that's about 1/3, that we'll see that later. One sort with MLL involvement, and that can be either a partner of 11q, and then the other ones, none otherwise specified. So these were three types defined at that time. And because we didn't know where to put them, we put other leukemias, including NK, the very rare NK leukemias, in the same chapter of the WHO. But what you must remember is that this is the important part today. So at the beginning, the EGIL, the European Group for Immunophenotyping of Leukemia, devised a scoring system that was not to be used for lineage assignment as it was designed, but I know that many of you are using it for lineage assignment. And we proposed a score that was based on the specificity of differentiation antigens for different lineages. And then you can calculate the score for each lineage, and to have a BAL, at least two lineages will have score higher than 2. In variant area acute leukemias, co-expression with a score lower than 2 is not rare, and you must think that these are variant types of leukemias. But for MPAL, it's more than 2 in more than one lineage. So this is the first classification that we propose, with strong lineage associated markers that carry 2 points, some with less specificity that carry 1 point, and then 0.5 points for the other markers that you can see here. In 2016, we modified a little bit the classification because the one that we proposed in 2008 was perhaps too restrictive. So now, if you want to define an MPAL and if you want to see that this cell is expressing markers for the myeloid lineage, the first one will be MPO. That was the only one that was retained in 2008. But then we recognize that it could be of monocytic differentiation. So you can use non-specific esterase and these markers of monocytic lineage. And in the case which is the most tricky, where you have weak MPO, it's not that easy to detect MPO as the antigen intracytoplasmic in your cells. Then you can rely on CD117, CD13, and CD33 to consider that you have myeloid markers on your leukemic cells. For the B lineage, CD19 and CD10 is usually enough. Or you can also add CD79 in the cytoplasm, CD22, or if you do it on bone marrow biopsies, you can stain for PAX5 and find the expression of this molecule. That cannot be done in flow. For the T lineage, cytoplasmic CD3 is really the hallmark, and it's the best marker ever, usually expressed without a surface expression of this molecule. So you have to look for all these molecules if you want to be able to detect MPAL, and of course you can rely on some other ones, as we will see this afternoon. In 2011, the EGIL performed a comparison with the 2008 WHO classification, and you see here that we could classify, we'll see that later, the MPALs in B and myeloid, T and myeloid, B and T, and B, T and myeloid. These are very rare. These are the most frequent ones. You can see here the characteristics of the patients. And what happened in the end is that only 9 cases would have failed being scored as BAL according to EGIL criteria. And very amazingly, also 9 cases would have been included by EGIL and excluded by WHO. So basically, it shows that these two classifications are very close and will allow you to identify properly your phenotypic acute leukemia. So I will show you now some examples. This is a very rare case of B and myeloid, where you will see here that you have MPO expression together with CD19 on this cartoon here. You also have TDT, and you also have some CD13 here and CD79A. So this is probably the most characteristic one. If you use a cytoplasmic tube, you will see what I presented before. Here you see the CD45 side scatter with this huge area of the Bermudes, which is actually the leukemic cells. You see a little bit of neutrophils remaining and some lymphocytes and very, very few monocytes. If you look at MPO against CD13, you will find that the MPO is clearly expressed in this case. And here you have the positive control of very bright MPO stained neutrophils, which also express CD13. Here you have co-expression of cytoplasmic CD3 in this pattern. And here you see that there's B cells here and no staining on the neutrophils. And again, the MPO without this B cell marker. So if we consider this pattern, the third one, and if we backgate that, we find that it's exactly the pathologic population, which is co-expressing MPO and cytoplasmic CD3. So that's a T myeloid MPAL. Here you have a case where you see that, again, the Bermudes area is quite consequent, and very few neutrophils remaining, some lymphocytes. If you do this type of display, which is called I always miss that, density plot, then you will see that the size of the population is quite consequent here for the blast cells. If you look here at CD33 and CD19, you will find that you co-express these myeloid and B lineage marker. You can see that again with CD13 here. And you can again have CD19 and MPO. MPO is sometimes partial, but you can see that it's really positive here. Again, you have your positive control of the neutrophils, which express the myeloid markers, and your lymphocytes that are not stained at all in this case. And that's a B myeloid case. BT is very rare. And you see here that you have co-expression of, I see, cytoplasmic CD3 and cytoplasmic CD79A. This case was from the literature. There was no MLL rearrangement, and this was published in 2004. You also have in some cases biclonal MPALs, where you have these two different populations, one of each lineage, and you will see that in the microscope if you look closely, because you will find that you have blasts of different sizes. But it's difficult to see on morphology, but you will see it in flow. Here in this display, you see that there are probably two different populations here. You have erythroblasts here, lymphocytes here, neutrophils, no monocytes, but you have clearly two different clouds of cells here. And if you stain for CD117 and CD34, you will find a population which is CD34 positive, 117 negative, and another one that is co-expressing these markers. If you look at CD19, you will find that the purple one here is expressing CD34 and also CD19, while the other one does not. And if you look at the backgating of that, you see that precisely your two clones are the two clones that you could see there. So one is B, one is myeloid. And that's a biclonal B myeloid leukemia, MPAL. And you can also use the radar display to show that these two clones are really definitely distinct and separated by their size and also by their markers. So what's the natural history of BAL at the time? Because there's a paper that Stella Matutes published with us in 1997. The incidence is less than 5% of acute leukemia. So you see that it's very, very small population. It will have AML morphology in about 2/3 of the cases, L1 or L2 according to FAB in the other cases. And as I said before, there are 4 immunophenotypic groups: myeloid B, myeloid T, myeloid BT, and BT. Usually these cells express TDT, CD34, and HLA-DR. That's not going to give you much of a clue, but it's a feature. And they will have no specific chromosomal anomaly, but I showed you before that about 30% of the cases will have a Philadelphia chromosome, and that there was a frequent involvement of 11q23. The clinical features is not very typical. It can occur in infants or in elderly patients, so over the whole age range. They can be de novo or they can occur at relapse. They usually have a higher white blood cell count, but not always. And the major feature is that they have a poor outcome. They express PGP, so that they're going to be very chemoresistant because they expel the chemicals, the chemotherapy. And in the literature, most of the cases report various isolated anomalies, and sometimes the definition is limited. So it's not that easy to make a meta-analysis and have a real good idea of the incidence of that. But there was a survey that produced more data, the SEER survey, that showed that it was 2% of acute leukemia, so well under the 5% mentioned by Estella. Incidence of 0.35 per million inhabitants, no specific age range again, AML morphology again in 2/3 of the cases, and clinical features of classical acute leukemia with fever, fatigue, bleeding, infections, all that related to the involvement of the bone marrow. High or very low sometimes leukocyte counts and bone marrow infiltration. And in some isolated cases, lymphadenopathies have been reported, or even a few cases of extramedullary, extranodal disease, like for instance, skin involvement. Here is the outcome of the T myeloid and B myeloid, so much below AML and ALL. This was some time ago. Now the ALL curve is certainly much better. And if we go to more characteristics, you will find a paper in Clinical Cytometry Part B that was published last year, where we were able with Anapoid to make a meta-analysis of 723 cases that we could retrieve in the literature. And from that survey, we found that the cytogenetics was, karyotype was normal in 36% of the cases, complex in about also 30%, involvement of KMT2A MLL in 14%, Philadelphia chromosome in 13% only, and hyperploidy in 9%. So it's not exactly what was reported in the papers that I mentioned before. It's probably more representative of real life because the number of cases is quite high here. We could retrieve molecular data in 180 cases, and you will see that we found anomalies of Notch, RUNX1, FLAG3 in about 30% of the cases, SXL1, TP53. So these are things that you see in acute myeloid leukemias. This is something that you see in myelodysplasia. And of course, TP53 disruption is a very good marker of severity and this disappearance of this anti-tumor gene. Now, the question of the origin of MPAL can be tackled. Are they engaged cells? And if so, are they a diseased myeloid precursor? That went wrong. Of course, we can find in the literature cases that developed after myelodysplasia or myeloproliferative syndromes. So it could be that something went wrong in the myeloid cell. And we also find some chromosomal anomalies of AML, such as involvement of CBF in the A21 translocation. Or is it a lymphoid precursor that became aberrant? ALL morphology can be seen in about half of the cases, and chromosomal anomalies that you see in ALL, such as Philadelphia chromosome, but also 411 translocation, can be seen in some of these MPAL cases. A third possibility would be that it could be an early progenitor that is not definitely committed, something that would be hesitating between being a myeloid cell or lymphoid cell. So it could be the case, for instance, for the myeloid BT, very rare cases that we can see. And if you look at this paper of 2017 by Velten and al., it goes with this last hypothesis, but in a sort of normal way. What they did is that they singled out the cells and they did single cell transcriptomics and then cultures of the cells to see what kind of lineage would grow in the laboratory. And they found that there was, in fact, a continuum of differentiation, which can be represented like that. So you will have here a population that is not committed at all, and it can then move to megakaryocytes, erythrocytes, neutrophils, monocytes, or B cells, eosinobasal. So this sort of star is the direction that the cells can take from this pool here. And what they call that was a cloud HSPCs, a continuum of low primed, undifferentiated hematopoietic stem and progenitor cells. That's the acronym here. And you can find again these different stars with the cells going one direction or another one, and you see that it's related to the expression of specific gene patterns. And they summarize that in this cartoon, where this uncommitted cell is here and can hesitate at one point between what it's going to be in the end. Probably erythroblasts and megakaryocytes are in a different pathway, but here you will find that you can go to the lymphocytes, monocytes, or neutrophil pathways. So this cell or this one could be the one that is blocking its maturation and giving rise to MPAL. There's no proof of that, but I think that this paper is really worth looking at because it sheds a new light on this kind of differentiation patterns. So what's the therapy of MPAL? I told you that it was a severe disease and that the outcome is very poor. So the best thing that you have to do is to look for a donor as soon as you can when you diagnose this. And I recommend that you read this paper by Volash and Stone in 2015, which is really giving a good algorithm. That's the algorithm that they published. So the first thing you have to look for is there a Philadelphia chromosome? If yes, then you're going to use acute lymphoblastic leukemia like chemotherapy, so an ALL treatment, and tyrosine kinase inhibitor. If your patient and the ALL-like is pediatric inspired, that means with a pre-phase of corticoids and then a chemotherapy that you can do until your patients are 40 years of age. Depending on that's what you will do if your patient doesn't have Philadelphia chromosome. So it's basically the same therapy, but you will add a TKI for the patient with the Philadelphia chromosome to block the kinase. Then if the patient doesn't reach complete remission, then you would use an AML-like pattern with cytarabine and methoxantrone, for instance. You will have to reassess the phenotype and you will consider this AML salvage. If the patient doesn't reach CR, then you will try again a clinical trial or targeted therapy. And you can, if it doesn't work, go to allogeneic stem cell transplant. That's going to be the case for most of the patients, although there are now a few reports of patients who didn't need to go to allogeneic stem cell transplantation, especially for the cases with Philadelphia chromosome, where the tyrosine kinase inhibitor was efficient. So in summary, MPL practically is that we have little information given by the morphology. It can be lymphoblastic, myeloblastic, or undifferentiated. Immunophenotypic identification is necessary. If you can do a bone marrow biopsy, do it and use immunohistochemistry if you have this material available. And don't forget to use PAX5 because it's going to be useful in that context. It's simpler to find them out with the new classification that was published in 2016 in the new WHO book. So you can use the intracytoplasmic tube with MPO, CD19, cytoplasmic CD3 as an initial hint. You can also use CD79A here in this tube, but it's not enough. You need to have more markers to make the proper identification. You can, in a way, rely on karyotypic anomalies, especially for the treatment by TKI that I just mentioned. And you have to keep in mind the therapy algorithm with the fact that you will, in most cases, need to have this allogenic stem cell transplantation. And that's one of the first things to do is to look for a donor for your patient. So with that, I wish to thank you and I'm open for questions. I will just give you two little information. You can find this book that we published with Anna Porwit a year and a half ago, which will give you hints on flow cytometry in the diagnostic of hematologic malignancies. And this is the cytometry issue of last year, which is giving you this review on EMPA. Thank you very much.
Thank you, Marie-Christine, for a very interesting talk. So I will start with a question. It's a very difficult question for us, as we have a lot of samples, really a lot of samples coming to our lab. We have not very few samples with small leukemic population, additional small myeloid population, which is definitely leukemic. But the question is, from what size of this population it should be named mixed phenotype acute leukemia? I mean, if you have 5% of this leukemic myeloid blasts and a huge population of lymphoid cells, if you have 10, 8, 2-3% of these myeloid leukemic cells, and you have 50, 60, 90% of lymphoid cells, can you name this mixed phenotype acute leukemia, or you have to just mention that you have you find this additional leukemic population? What is the algorithm of describing of two populations?
So one thing that you have to keep in mind is that when you get bone marrow, it's in most cases hemodiluted. So the size of the blastic population is not that important. What you have to do is really to have an integrated diagnosis. That's a notion that I really favor. You need not to rely only on morphology and flow. You need also to look at the karyotypic features. You need to look at the molecular features of the patient. And mostly you have to look at the clinic of the patient. And any size of cells that are looking suspicious should be considered and treatment applied accordingly. Especially what you have to be careful is that if you find a leukemic population of lymphoid lineage and some myeloid cells beside that, you must be sure that it's not some normal hematopoiesis of the myeloid lineage remaining in the bone marrow. Because if the infiltration of the bone marrow is not that heavy with the leukemic lymphoblastic population, you can still have some myelocytic maturation, and you must be sure that these are normal cells. Usually you will see that because these cells, if they are really stem cells, engage the myeloid lineage, will be have a very low side scatter and will be very focused on your scattergram, 45 side scatter scattergram.
Oh, thank you. And the second question, maybe mainly regarding one strange case we had previously. Have you seen ever the situation of cytochemical myeloperoxidase expression, total cytochemical myeloperoxidase expression in phenotypically absolutely lymphoblastic leukemia with only some kind of 5–7% of cells positive, weakly, very weakly positive for myeloperoxidase in flow?
That's the question that comes very often, because it's not that easy to find out MPO. I will show you examples this afternoon where it's not easy to decide on that. In most of these cases, you will find other myeloid markers. You will find 117, or you will find 13 or 33. So you must again rely on more than one marker. And another thing that must be pointed out is that sometimes you find MPO in cytochemistry, because you see the activity of the enzyme and you don't see the antigen because the molecule has been modified in a way, or you can have the other way around, where you can see MPO in flow and you cannot see the enzymatic activity in cytochemistry. So I think that it's good to have both techniques done if you still do cytochemistry in your lab.
And how to manage the situation, for example, in which you have 4 or 5% of blasts cytochemically positive for MPO, even weakly positive, because in this case, in WHO, it is written that you have to find MPO by any type of methodology, and so you have, for example, the same. I don't know, 4 or 5% of cells myeloperoxidase positive by flow, but for flow this is below the threshold. I mean the threshold of positivity for any type of classification, and by cytochemistry it's above the 3% of threshold positivity. And how to manage this time of discordance between two methodologies?
Well, the threshold that is recommended for cytochemistry is 3%. In flow, it's more recommended to rely on other cells that are present in your tube, and mostly to look at the lymphocytes. And there was a publication a few years ago that used that, using the lymphocytes as the reference, and that said that 11% was going to be significant. But mostly you have to look at the pattern. You have really to make your monoparametric or biparametric histogram and see the superimposition of both types of cells and decide whether it's shifted or not. It's again, it's not easy.
Thank you.
Александр, сейчас я вижу в чате, что попросили прокомментировать ответ и вопрос, поскольку не совсем верный перевод показался. Я так понимаю, на русском языке просят продублировать.
Да, я попробую сейчас перевести. Это на самом деле ситуация, которую я озвучил, не самые простые. Первый вопрос мой касался того, в каком случае вы должны описывать вторую популяцию как отдельную популяцию, в каком случае вы должны описывать МПАЛ, ну, смешанный фенотипический лейкоз, именно как смешанный фенотипический лейкоз, если у вас, допустим, маленькая вторая популяция, чаще всего все-таки миелоидная, если она там 3, 4, 5, 8 и так далее процентов. Да, Мария Кристина сказала, что, в общем-то, нет в данном случае разницы, сколько процентов этой дополнительной популяции, поскольку образец костного мозга может быть разведен кровью, и это действительно так, мы получаем образцы костного мозга, которые разведены кровью, и это может быть, это может приводить к искажению истинного процента бластов, поэтому если вы уверены в том, что это опухолевые клетки, а не нормальные какие-то миелоидные клетки, которые могут быть вокруг основной лимфоидной популяции, ну тогда они должны быть в любом случае описаны. Второй мой вопрос касался того, возможно ли ситуация, допустим, когда абсолютно лимфобластный лейкоз с очень маленьким процентом МПО-позитивных клеток, там что-то в районе 7-8, в итоге оказывается химически тотально МПО-позитивным. Мария Кристина ответила, что так бывает, что, как сказать, бывает очень редко, это не так просто интерпретировать такие данные. На самом деле достаточно часто в таких ситуациях бывают еще какие-то миелоидные маркеры, да, что поэтому, в принципе, в таких случаях при помощи дополнительных каких-то миелоидных маркеров можно скорее понять смешанных фенотипических лейкозов. Нет. Ну и, наконец, третий мой вопрос касался того, что пороги для пероксидазы цитохимической и пороги для пероксидазы иммунофенотипической, они разные. И, соответственно, если у вас есть там 4% клеток с цитохимической пероксидазой и те же самые 4-5% клеток с симметрической пероксидазой, формально результат будет разный, да, поскольку в одном случае у вас будет больше 3%, больше того порога, который введен для цитохимического определения пероксидазы. В случае для симметрической пероксидазы такого порога нет, но она тут, собственно, сказала, что пороги симметрические вообще разные. Действительно, была в свое время очень интересная статейка, на которую она сослалась, по выявлению разных порогов при калибровке частицы, калибровке клетками, да, для того, чтобы понять, где начинается истинная позитивность миелопероксидазы, и, соответственно, был предложен порог в 11% при использовании в качестве негативного контроля лимфоцитов. Это очень интересная такая разработка, действительно, я в свое время читал. Ну и, соответственно, да, пороги разные, но в данном случае, есть некий стандартный тип ответа о том, что надо все анализировать в комплексе и думать обо всем вместе, что это действительно сложная ситуация, которая требует дополнительного размышления. Как-то так.
So I'm going to give only a live session. I'm not going to make a presentation before that. And the agenda is that I've collected 8 cases, and there are various panels from 4 to 10 colors, because as I said this morning, these cases are rare, so I had to go and dig in the archives to find old cases. And I also got 2 cases from Anna Porwit that I will show at the end. We will start from scratch for the first case, although you know that I would just like to show it in case some of you are not so familiar with the template or protocol creation. Then we will adapt from these initial templates, and I will give you some information about the patients as we go. So let's go. So this is, don't be afraid by the list that is on the left. We're going to go through that properly. So we start with the first case. The first case is an old lady of 84 years old, and she was diagnosed with this blast in the peripheral blood, and then we went to the bone marrow. So what we first do is a forward scattergram to remove the debris. And then now I was engaged in making this first gate. Never works well when you're trying to demonstrate. So here on the left is the fat in the bone marrow that I'm going to remove. And here we are. And if you want, for diagnosis, it's not important, but if you do MRD, it's important that you also get the cells that are stuck over there on the top, so that you really take all the leukocytes from your sample. Then we make a 45 side scattergram, and we remove the color here, away. And now we're going to enlarge this one to make our gates. Of course, we first get that on A. And then these are our polymorphonuclears here away on the top. I think these are true, and there are very few monocytes in this sample. So now I do the monocytes. Thing like that, not very important here. And that is monocytes. Here we do the PMNs. And now we have to do the lymphocytes. And my preferred magenta. I noticed that also Michael is using the same colors all the time, and you will find that this is very useful because then you, as I said this morning, you will recognize your population straight away. Then you choose the colors that you like, but it's good to stick to them once you've chosen something. So here I make a B gate that is going to help me to get the Bermude Boolean gate. And I think that I have kept it there. So that's PMN, oh, I missed the B. It should be B. So I must do it again. There's going to be B and not PMN and not MN and not LY. So now we have our Bermudes, and it's already set in my computer that it has to come in this blue, sea blue color, cyan. So now we're going to make plots for the other things that we have in this tube. And I like, as Michael also showed in, with his colleague, that I like to have each fluorescence against side scatter because it makes it very easy to find out what's going on in your sample. So I'm going to get that on A. Actually, I could remove a little bit of this rubbish here, but that doesn't really matter. And now I'm going to copy this one. So I have CD10 in this tube, I have CD22, I have CD19, and I have CD38. And what I see now is that, What I have to look at now is this blue population, because this blue population is the one that is our blast population. We can also try and make a density plot if you want to be sure that your gates are properly set. And you see here that there seems to be two populations that you didn't see at the beginning, but we let it with the color. And we see that here we have CD22, which is clearly sticking out from the blast population. We have CD19, and we have CD38, which is any kind of lineage, but clearly we are in a B lineage here. So let's move to the next tube. So we keep our template. Now we've done it for the whole session. And we see that we find more B markers, CD24 here, CD19. You see that there are a few B lymphocytes remaining there. And then you have your CD34. So we're still in the B lineage. And now we see that we have 117 that's coming out. And probably if we make now another biparametric histogram, we will see what happens. So I will put CD19 here and CD117 here. And it clearly is two different clones, as you can see. I can make it clearer if I just gate it on the Bermude. And I can also use this logical function to see better my populations. And you clearly see that you have these two different populations, at least perhaps even three, one CD117 positive, one CD19 positive, and one without any of these markers. So this is a typical case of biclonal, two different clones coexisting. And if I move again with my colors, if I make new colors for these two populations, I will find out that this, in fact, mimics what I saw when I did the bandit coloring, the density plot. These are the two populations which are really showing that we are in a biclonal population. I can go and look at whether there are other markers. And here I find out again that CD19 and CD33 are in these two different populations, these two different clones. What I want to do is this, go back, and then you see again that these are two different populations. And I've got one last marker, which is an important one. It's a CD79A in cytoplasmic, which is positive. And I have the MPO in another tube here. and the MPO is negative in this case, but it's typically a biclonal population. And the B1 here is definitely MPO negative. That was the first case I wanted to show you. So now we move to another one. Maybe I remove this because I'm not going to have this situation. And now I have to change my gates. Of course, this second case is, he's a man of 64 years old, and he was diagnosed with this very dense population of abnormal cells. So I make my boolean here. There are very, very few polymorphonuclears there, and I think that my gate is okay like that. I can check a little bit with this formulation. Yeah, that's okay. So I can go back to the colors and try and see what I can find out in this population. So the cells, part of them are expressing CD7. They are clearly CD13 and CD117. There's nothing here in this tube which has less markers. That was, I think, a 4-color tube. Yeah. So we have 1317 and CD7, which is not rare when you are looking at AML. And as I showed this morning, it's also something that is physiological. There is a small population of normal growing cells that co-express CD7 and myeloid markers. So we go on and we have CD33, so we're still fully myeloid, partial expression of CD34, some CD36, and part here of CD19. You see that there are very few normal B cells here, purple, but part of the blast population is expressing CD19. We also have some CD4, which can be seen also in myeloid markers. So it's not going to be a BT myeloid. So far, I would guess it's probably going to be a B myeloid. And this will be shown here because you can see that if I make a gate around my CD34, sorry, my CD79, they are expressing MPO just the same way as the other cells. So it's a weak expression of MPO, but it's clearly the same population that is this time really co-expressing myeloid and lymphoid marker. So I forgot to tell you that the first lady that we saw, she was treated with an ALL protocol, and she's doing well. That was a case when she was then receiving perinatal and methotrexate, and she has been actually enjoying quite a long complete remission. It wasn't so good for this man. This one, he received first an ALL protocol, and which was not efficient. So then he was moved to a myeloid protocol with midostaurin, and then he received an allogenic stem cell transplantation, but he had complications at the end, and finally the story didn't end well. Now we move to a third case. This one is also a man, 58 years old, and here's what we found. So again, quite a very neat population of blasts. These are diagnostic bone marrow, so as was said earlier on, it's always easier on the diagnostic. You don't have to make, unless you want to prepare for the search of different for normal or LAIP. Here, it's not the same strategy. We're just trying to see if markers are co-expressed or not. So here we go. And what do we have? Here we have CD7 clearly expressed, brighter than in the previous case. We have no CD13, no 117, no 14, but we have 33 and 34. No 36, no 15, no 19. So far we have one T lineage marker and one myeloid marker. Here we don't have MPO. We have probably an abnormal, but sometimes these antibodies are sort of giving this not nice picture, but we are clearly CD3 positive intracytoplasmic. So we have intracytoplasmic CD3 and CD7. That's the phenotype of a T1 ALL. So we go on to see if it's really a T1. So we see that there's no surface CD3, that's only the remaining lymphocytes. We find a little bit of CD5, not too bright, no CD8. No CD2, no CD16, no CD1A, no CD10, and some HLA-DR. So if we make the sum of what we have a phenotype of a T1 with abnormal expression of CD33. As I said this morning, that's not enough to make it an MPAL. So in my mind, it's a very early T cell progenitor ALL. And in such cases, it's not rare to see the expression of CD33 or one other myeloid marker. So this one would not be considered like an MPAL. The patient received the treatment for ALL, classical protocol for TLL. He also had FLT3-ITD mutation and mutation in TP53. He didn't respond to therapy, so he was moved to Vidaza, which didn't work. And then he was in such a bad shape that we stopped the treatment. So let's move to the next one. The next one is wait a minute, MPAL 4, is a 40 years old man. And here is what we had. So we need to make some adjustments here. Oops. And here, this is probably going a little bit on the left. We can make this window okay like that. So probably have monocytes here. So I make this bigger to see better what I do. Like that. So because of the Boolean, it doesn't matter that my monocyte gate is going over in the box because it's going to be taken away by the Boolean equation. Same here, and my lymphocytes are a little bit on the left here. And I have some dark blue here because of this thing here. So what do we have? We do have indeed monocytes, which are stained here in green for CD13. We have also part of the neutrophils that are like that. And of course, we have most of the blast gate. And if I do that on Bermuda, I will see that better. And indeed, my population is very clean here. I think I've lost my Boolean. No, there's something that's not right. Oh, yes, it's too big here. Here we go. Okay, and we do also have some 117. So I do it on the Bermuda again, it's cleaner. So that's what we have, 13, 17, so we're moving to myeloid there. It goes well because we now have CD33. You still see your monocytes in 14 and no staining on the blast. CD34, no surprise here. No 15, and we have a little bit of CD19, which is different from the normal B cells, because if I go again on my A gate, you see that there are very few B cells. Most of my lymphocytes are T cells, and clearly this is a population, subpopulation of the blast population that is expressing 19, but very a minority of the cells. I find that this population is also 79A intracytoplasmic, so it still goes with that. And I see that there is some MPO on my population here. And I guess that if I do, so there's no CD3 as you see here, and if I do my CD79 versus MPO, I can see this is one part of the population, but you see that the CD79A is only weakly expressing MPO. So we are really this is actually, this is part of the neutrophils, as you will see here, because you see here that the bright MPO in the neutrophils, and we have the same intensity in the 79A and the myeloid cells, which is slightly bigger than what you have in the T cells here. So this case could be classified as a biclonal B myeloid, and this patient, wait a minute, that I find my paper again. He was treated like a B1 first, and then he escaped. And what happened is that he was treated like for an ALL, and he got all the population of the B cells removed. He evolved into an M4 myeloid leukemia with no sign of the B cells remaining. Then he was treated with an AML protocol, and he received an allogenic transplantation. And he relapsed, and at relapse, we saw exactly the same pattern as at diagnosis, with, again, this population of CD79A with and some MPO on it. So that was a very bizarre case. So the next one, we have to clean that up a little bit. So we're going to make that bigger. So some neutrophils here. Lymphocytes are down there. Up. We have perhaps a few monocytes around here, not too well. And here I have my population of blast cells, which is slightly with a very low size scatter. So I'm going to remove this gate again. And so what do we have here? We have clearly CD7 on the blast cells. We have some here side scatter. So we have CD13, so 7 and 13, and 117. No 14, no 33, 34, not very informative. 15, another myeloid marker, no CD19, no MPO, and a very nice intracytoplasmic CD3. So this CD7 that we saw was probably related to this population, and it's co-expressing the myeloid markers. And it's also CD2. So we have the number of T cell markers that we want, 27 cytoplasmic CD3, and we have the myeloid markers. So this was a real case of T myeloid. This was a young boy, I didn't tell you before. He was, I think it was 17 diagnosis. He was treated with an ALL protocol, plus midostaurin because he had a FLT3 ITD mutation. He was in complete remission, so he was given an allogeneic stem cell transplantation, 10-10 pheno-identical, and now he's nearing his second year free of disease. So that's really typically the case when you must look for a donor very quickly and do it as soon as you reach a complete remission, which was the case for this young boy, and we hope that he's going to keep doing well. So now I will move to another case, and this one is this one is a woman of 50 years old. She had a Philadelphia chromosome, and what we found was this. Let me adjust a little bit. So very few lymphocytes in this bone marrow, probably no monocytes, and here we have the blast population here that we can also show like that. Quite nice, only single population here. So what do we have? We have some CD7, CD13, 117. No 14, some 33, a little bit of CD34, partial expression. CD36, and now we have CD19. So perhaps the 7 that we saw on the first plot was something that would go with the myeloid profile because it was only partial. And then we see here that it's clearly part of it is CD19. And then if I go to this one, I will find very tiny MPO, but I will find again my CD79A intracytoplasmic. And here I have another tube that is allowing me to check for the co-expression of the markers. So I go to CD45, it's a different one. So everything is coming in order. I have to check my lymphocytes because the tube is not the same and the settings are not exactly the same. Let's make this window a little bit bigger. And then I see the 117 and the 19. And if I make a combination of that here, you see that there is this sort of continuum. So these cells here are clearly not 717, but they go on to reach 17, and these cells are going down there. So this is a slightly heterogeneous population, but with a clear co-expression, at least for this part of the cells of the CD19 and the CD117. The story of this patient was that she was treated with an ALL protocol and then with a myeloid, and she relapsed. At relapse, she still had the B compartment, so she received blinatumomab, but then her situation worsened and she finally died from her disease. So these are cases from none for which I had the history of the patient. You know that you've seen that it's not really a good prognosis. Only this young boy is still alive in this population. And now we'll show you 2 cases from Anna Powitz. So she has a 10 color tubes. We have to make some adjustments here because it's a different protocol. She also works on the Navios, but she uses different parameters, and we will have a more complex population to look at. Sorry, side scatter. So here we have population. Maybe we can clean it a little bit. No, it's okay. Just maybe that. Okay. Now she has, of course, a different 45 in chrome orange, and the side scatter int, side scatter linear, and the rest logarithmic. So let's see at what we have here to make these gates proper. What we can do is 2 things that I wanted to do. One is to only show part of the population because it's going to make things easier. So maybe I will make 10,000, maybe 20. And then I will also use this banded coloring to see where my populations are. And indeed, this is a population of blasts, which is almost evoking an M3, but not really. M3 is really looking like the polymorphonuclears have fallen down. I guess that these polymorphs are something like here. We'll see afterwards what's going on. Now a few lymphocytes here, and perhaps some monocytes here, but I'm not sure. Something like that. So let's see what it looks like. So I'm going to make my markers here. So FITC, and of course I will use side scatter int, and it's easier to go like that. And then to copy this one. And then we move our different markers. So FITC, PE is MPO. This one is CD2. This one is HLA-DR. Now we have some 19, 79A, CD34, CD33, and that cytoplasmic CD3. There's 10 markers. So what really looks like is that we might have 2 populations in this gate. So it looks like it's going to be biclonal. So how can I confirm that? I can, for instance, try and do the 19 versus 33, which looks quite promising. And indeed, I have 2 populations which are completely separated. And I use the banded coloring to see that really there are 2 populations very well defined. I'm going to put it in logical. It's going to be nicer like that. Okay, can skip that a little bit. And clearly there is one population here, which is very weakly expressing 33, but which is B, and another clone here that is the myeloid one. And funnily enough, you don't really see these 2 different populations here when you do the density plot. So let's try to make some color to see where they are. So if I put that back into the color gate, and if I make a box around my myeloid compartment here, I see that it's almost everyone still. And if I make my box here, and I see that it's this tiny population at the bottom that seems really clearly separated from the other one. There might be a continuum, but really from this picture here with 19 and 33, it really clearly looks biclonal. I can also get that up a little bit to see my populations better. And I might, again, as in the first case, be a small population of nothing at all in that, but it could be some erythroblasts that are around there. So I don't know the story of this patient, nor do I know the story of the next one here. And now we have the proper template because it's the one that I adjusted for Anna's tube. So just a little bit debris if you want. very few other cells than the blasts in this population. So I can make my box a little bit smaller, maybe a few monocytes, lymphocytes, very few on the right here, and polymorphonuclears, we can get that down a little bit, but it doesn't really matter. So what do we have in our Bermuda area? We have TDT, we have MPO, which is quite clear in this instance. We have CD2, we have HLA-DR, we have no 19. I can put that in the proper color and remove my 33, just to see with the side scatter fluorescence how it looks. So no 19, I can remove that gate too. No CD79, I think that's again a sort of wrong labeling, but I see clearly cytoplasmic CD3 and CD34. So it looks like we have a myeloid with MPO labeling here and cytoplasmic CD3. And she didn't give me the rest of the tube. I think there was some CD2. Oh yes, we have CD2 here, we have cytoplasmic CD3, and we have MPO. So what we are missing in this tube is more myeloid markers. I guess that Anna has another tube and that she could confirm that it was a T myeloid MPAL. So that's about what I wanted to show you. I hope I wasn't too fast. Everything is still there, so I'm going to ask you if there's any question that you would like to ask me. And I think that after that, you go to another break, I believe.
Christine, of questions about what do you think about TDT and 79A? What do you think about marker TDT?
I myself, I'm not too fond about TDT. TDT is mostly interesting when you want to make the difference between lymphoma and acute leukemia, because normally lymphomas would not express TDT, except sometimes DLBCLs, but it's not going to provide any help. It was a 0.5 point in the EGIL classification, and it can help you if you want to be sure that it's really immature, but that's the only thing that it helps for. Okay, the questions are all in Russian. So I took the cases I had. It's not a demonstration, it's really real life. It is sometimes a little bit difficult to be sure what's going on in there. But what was absolutely certain from the cases I showed you was that you had myeloid markers and you had lymphoid markers, except for the T1 case that I showed, which is expressing CD33, and we can expect that from these cases in normal ALL. But the other ones were all at least biclonal, and in most cases, really MPAL with a mixed phenotype that you could see in your population. So the message is really that you need the panel that is big enough to make it. It's not necessarily to have 10 tubes or 10 colors or 12 colors that we can do now, or even 13 colors if you have getting the new DXFlex. I showed you also that if you're using even less markers, you can still find out your population. And if you have the impression that you have myeloid markers and lymphoid markers, and you've done that in different tubes, then you can make another one to be sure and be able to show for at least one set of markers that you do have the co-expression or the two clones, which are different. So even if you do it with a few markers, you can still find out your population. The very important thing is that you use enough markers to find them, because as you saw, the outcome of this patient is very poor.
Okay, we have one more question. Have you ever seen linear TB myeloid leukemia, acute leukemia?
Myself, no. As I showed this morning, I had to take that one from the literature. But you know, one of my sayings, people in the lab laugh at me, but I'm always saying that cells are doing what they want, and that we have to look at what they do and we do not have to force them in a box. So anything can happen. The genes are all there for the expression of anything on these cells.
Thank you, Christine. We have no more questions. Only thank you very much from our.
Thank you, and thank you for Sergey, who wrote in English to me, and also other people I can't read the name because it's in Russian. Enjoy the end of your session, and I hope that I brought something to you. If you have any question that comes afterwards, you can always ask for my email address, and you can send me a message. I will answer. Bye-bye.